それでは時間となりましたので、セミナーを開始いたします。本日座長を務めますのは、九州大学アジア・オセアニア研究教育機構のキム・シューマッハ准教授です。シューマッハ先生、よろしくお願いいたします。
Thank you very much, Anai Sensei, and welcome everyone to the 158th edition of the Brown Bag Seminar hosted by QAOS, the Kyushu Institute for Asia and Oceanian Studies. And today we are looking forward to hearing from Associate Professor Vishwajit Chowdhury, and he will be talking about a very relevant topic, I think that we can all relate to: summer heat stress. Beat the heat. And Professor Chowdhury is part of the Lab of Animal and Nutritional Physiology at the Division for Experimental Natural Science at the Faculty of Arts and Science within the Graduate School of Bioresource and Bioenvironmental Sciences at Kyushu University. He has been providing education also for Kyoso Gakubu at the Graduate School of Bioresource and Bioenvironmental Sciences, and has been researching about stress and nutritional physiology on top of that. In terms of achievements, he has been the recipient of the Japanese Society of Animal Science Award in 2022, and also the Japan Poultry Science Association Award in 2017 for his research on heat stress in chickens. Before joining Kyushu University in 2010, he was working at the Tokyo Medical and Dental University in the Department of Biology as an adjunct associate professor in 2009, and he was a lecturer of biology at Hosei University in 2008. He worked on novel neurohormones, gonadotropin inhibitory hormone, and its homologs at Hiroshima University and Waseda University from 2004 to 2008 as a postdoc and a research associate. Professor Chowdhury has authored more than 100 peer-reviewed original papers, 13 review papers, and 3 book chapters, as well as numerous symposium articles. And Professor Chowdhury achieved the President Award when he obtained his PhD degree in 2004 from Hiroshima University for his research achievements on stress physiology. With all these credentials, we are very much looking forward to his presentation. And without further ado, I would like to invite Professor Chowdhury for his presentation. Thank you very much for being here today, for your time, and the floor is yours.
Thank you very much, Professor Simha, for your kind introduction to me. And I'm highly honored and privileged to get this opportunity to talk here in front of knowledgeable and general people as well. So let me share the slides now. So I will talk about heat stress matters, focusing in chickens, but which may be related also for humans as well. So let's talk about today's topic. So first of all, climate change. Everyone nowadays knows the term climate change, and NASA has been doing an excellent job to collect different kind of data of climate change. For example, carbon dioxide level, atmospheric carbon dioxide level, currently 426 ppm, which is significantly high, compared to the previous years. I will explain later more about it. Global surface temperature also increased. Methane gas also increased a lot, and then sea ice declined. If we explain more about carbon dioxide, then you'll see the graph like this. This is the x-axis indicates the years before today. So this is today here, and this is 800,000 years ago. The carbon dioxide level in the ice core data, these yellow lines indicate ice core data. At that time, modern instrumental technology was not discovered, but when it discovered in 1958, then that instrumental data is showing here in the red line. So if I explain simply, the carbon dioxide level in the millennium, 180 to 260 ups and downs, and maximum 300 ppm it was. But in 1911, it crossed the 300 ppm, and now today it is 426 ppm, a significantly high level of carbon dioxide. The carbon dioxide has a capacity to trap the heat. The heat coming from the sun is trapped by the carbon dioxide, so our planet is very hot, and particularly summer become scorching summer nowadays. You experience those who are living in tropical and subtropical area of this world. Everyone knows how severe heat stress was in this summer. So due to the climate change, many plants, animals in this planet are disappearing. For example, climate change caused to kill this golden toad. This golden toad is no more available in this planet. So with this climate change, different species of animals, insects, plants are disappearing. This is really alarming. In July, this is a photo from Shinjuku Station in Tokyo, and July 29, you see people are roaming around the station, beside the station, and using umbrella or a hat, or trying to protect them from the heat. So this year, summer month of June through August were hotter than usual, resulting record temperature in July, a trend that continued in Western Japan last month. I think you may remember that in 2021, in June, around 29, in Canada, British Columbia, the temperature suddenly increased around 50 degrees Celsius. Many men, animals, plants, agricultural system collapsed for a certain period of time, and it was so alarming. Then if we focus on chickens, chickens are very susceptible to heat stress because their body is covered by feather and their metabolic rate is very high, so their body temperature very high. Some of you may know 41.5, around 41.5 degrees Celsius body temperature. And they don't have any sweat gland, so they cannot sweat. Sweating is not possible for them. They don't have sweat gland. This is resembled with the older people. Those sweat gland is less active. And the young kids, they are developing sweat glands. So for the humans, older people, aged people or young peoples, this chicken can be a model animal of heat stress study. And then if we consider about commercial chickens, then the broiler or layer chickens, they are very susceptible to high temperature. During the heat stress, their body temperature increase, and then the food intake declines, their meat production declines, egg production declines. Many abnormalities happen, and in certain cases, they can die. So, but native chicken like Chowdhury in Japanese or some jungle fowls, they are more stronger against heat stress due to having some genetic makeup, as well as their metabolic rate is low. So, but this Chowdhury or native chickens are not so frequently used for the commercial purpose because the productivity is less. But here you see some chicks, they need additional temperature at the beginning of their life, 30 degrees Celsius for say. So people are thinking that chicks are not subjected to heat stress. But our study first revealed that even 5 degrees Celsius high temperature compared to the standard temperature, it causes stress to them. But anyway, the chickens, it is reported that in the world, there are many chickens, 20 billion chickens live on our planet. In 2015, it was saying like that. And if we distribute for every human, we have 3 chickens. This is good news, not only for consumption, but also taking care, responsibility we have. Then we see about the meat production, because we are depending on chicken meat and other meats. So here you see the blue portion is some beef and veals, then the gray portion is for pork, and the green portions for poultry, chickens, meat mainly, and some sheep, other things, red portion. So as you see, if you compare, then you see that with the progress of years, production of poultry is increasing, and it is dominating than other meat production. So it is a vital meat source for human consumption. And then human population is also increasing, and it is after 50 to 70 years, it is predicting, the prediction is that in Asian region, the population will increase more, and African region as well, compared to other parts of the world. So the increased populations need more food, and then Asian African countries, many countries are locating in tropical and subtropical zone, where heat stress is a serious problem and chicken production is under threat. So we need to develop new technologies to survive the chickens under the high temperature, to adapt with the high temperature. So chicken has another benefit, is that the genome of human genome organization is more similar to chicken than mouse. It was reported in Nature paper 1991, Bart et al. They reported it. So many medical study or biological study, they use mouse or rat because they're mammals and human is mammals. So many studies conducted with mouse and rats, but the biological rhythm is different, humans and rodents, because humans sleep at night, but mouse or rats sleep active at night. In contrast, the similar humans and chickens biological rhythm, light rhythm is same. So chickens can be a good model animal to study many human disease or human physiology, biology, many things. So from now on, I would like to share our research findings, and I will try to make it simple as possible. But we have some Q&A session later after my talk. So if you have any question, please feel free to ask me in English or Japanese, whatever you like. I'll try to respond to you as much as possible. So food intake, as I mentioned, food intake declines by heat stress. We use different days of chicks, 3 days to 21 days, and black bars are the control temperature, open by high temperature. You see by the high temperature food intake decline, and particularly 2 weeks and 3 weeks chicks are more susceptible to high temperature because they reduce more food intake. And the plasma or blood glucose is an indicator of stress. So during the stress time, high temperature, 2 weeks or 3 weeks old chicks, their blood glucose level is very high. This is similar to human. Human also, when you are exposed to stress, our blood glucose level increase. This increment of blood glucose to provide us energy to fight against stress or to flee away from the stress side. So this indicator of stress, besides some stress hormone. So this was the first study where we reported that chicks, which was neglected from long time, that chicks are not subjected to heat stress, but we found that 10-degree high temperature compared to the control, 4 hours, only 4 hours exposure caused this sort of stress. And this study was published in Journal of Poultry Science, and it was selected the best paper award. Later, I like to very simply point out some other research like brain hormone. One of the brain hormones is neuropeptide Y, NPY. This NPY, we found that due to heat stress, it increased in the brain and its receptors, body temperature increase, stress hormone increase. But when you injected NPY in the brain, then its receptor Y5 increased and body temperature declined through the NPY5 receptor, and stress hormone declined, as you see here in this figure. This NPY treated group, this control group, and black by high temperature, open by control temperature. So plasma corticosterone or blood corticosterone is a very well-known stress hormone, and this stress hormone declined by the NPY injection. So NPY in the brain working as anti-stress factor, and this work has been conducted by Azim from Afghanistan. He got recently PhD from Canadian University. He did masters in our lab. So Dr. Azim, and then Dr. Hatem from Egypt. He did a study with me in my lab, and he also published this work. Then recently, Dr. Nishimura, she completed her PhD in my lab in last March, and now she is assistant professor in medical faculty at Kyushu University. And she found that NPY receptor, particularly Y4 receptor, is located in the spleen. So spleen is a vital immune organ for chickens. So what kind of function is conveyed with this receptor? It is our future interest, because clearly it was showing that the Y4 receptor localized in the splenic red pulp. These are the shape of red pulps. So it may play a functional role for immune regulation or some other cellular function. We are interested to find out the functions of NPY Y4 receptor in the spleen in our future study. Then another study is that when we injected NPY in the brain, then a taurine amino acid increased. I think some of you know that taurine drinks, sometimes you take sometimes taurine drink, which is good for health. We know the taurine, when we put in the brain, then it reduces body temperature. And we found that the brain's hormone, like dopamine, serotonin, you know, the good hormone increased. And then when we use some inhibitor of those hormone production or hormone synthesis, then we found that the body temperature reduction was disappeared. No body temperature didn't reduce. So it does indicate that so dopamine, serotonin, those sort of things in the brain regulating body temperature, because in our brain, we have the center of the body temperature regulating center is located in the brain, particularly a particular paraventricular nucleus, anyway, in the center of the brain, the hypothalamus area. So this study was conducted by Dr. Zakaria. He did also PhD with me and recently came back to my lab as a GSPS postdoctoral fellow for 2 years. And he published this work in European Journal of Pharmacology. So now we'll talk about citrulline, which is the main focus of today's talk. We found that the citrulline is the chemical structure of citrulline. This citrulline was first discovered by some Japanese scientist, Koga and Otake, in 1914, and Wada, 1930, he identified the chemical structure of citrulline first from watermelon. And watermelon, the scientific name is Citrullus vulgaris, from where the citrulline name derived. So this citrulline, we have found that when you expose the chicks to high temperature, the red bars indicate the high temperature group, their citrulline plasma, blood citrulline level decline. And this study was reported in the American Journal, Comparative Biochemistry and Physiology. We didn't know that time why citrulline declined and by the heat stress in the body. So to know the function, physiological function, we then wanted to know about their function if we inject in the chickens. Then this is the metabolic map of citrulline. Citrulline and aspartate produce arginosuccinate and then arginine and then ornithine. Ornithine go to mitochondria. This is the metabolic pathway for chickens. And then nitric oxide is produced from the arginine and byproduct citrulline is produced. Nitric oxide has many functions like body temperature regulation, blood pressure regulation, neuronal function. Many functions can be gaseous substance. Is a very important gaseous molecule. So we injected citrulline, arginine, and ornithine, these three amino acids, to the chicks, and we wanted to know their effect on body temperature. This is the result, and you see compared the control, the open bar, citrulline, this is the triangle bar, citrulline reduced body temperature within this 2-hour study in chicks. Then we used different doses of citrulline, and we found that high dose significantly reduced body temperature. So then we wanted to know what is the mechanism of reducing body temperature. So maybe nitric oxide may be the reason, because nitric oxide can regulate body temperature. So use inhibitor of this enzyme, and then we thought that, hypothesized that nitric oxide would be declined. So, but our result was different in connection with our hypothesis. Citrulline didn't change the blood nitric oxide level, but LNAM, the inhibitor, can reduce. So citrulline reduced body temperature, but didn't influence nitric oxide. So citrulline-dependent reduction of body temperature may not be connected with the nitric oxide. Some other factors would be relevant with this reduction of body temperature of citrulline. effects. So anyway, this is some normal data which is showing that the rectal temperature declined until 5 hours. We studied with a single dose of citrulline. It was 15 millimole dose, almost like 3 gram citrulline, and some other data here. So then we wanted to know what about the metabolomic status, overall metabolism of the chicks. Then we injected orally citrulline and then found that within 1 hour compared to the control, body temperature significantly declined in the chicks. And then principal component analysis also showed that compared to the control, citruline's expression different, and heat map also showing that citruline's expression different than the control group. So citrulline has some specific function in the body. These are metabolic functions of the citrulline-dependent metabolites in the TCA cycle. I'm not going to explain details, but simply I'll try to explain. When the citrulline is administered to the chicks, the blood citrulline or liver citrulline, sorry, there is a liver data, liver citrulline increase, and the subsequent metabolite increase, arginine increase, ornithine increase. So ornithine can go to mitochondria, and arginine also go to mitochondria. The mitochondria is a powerhouse, as you know, it produces ATP. So we see that with this increment, the red bars indicate the citrulline treated and water injected group are blue. So you see the NAD, NADH plus, everything is increasing with the citrulline administration only within 1 hour of treatment. So these molecules can produce ATP. ATP is storage energy of our body. For example, now I'm talking, so I'm breaking my ATP and I'm talking using my ATP. And you are listening, giving your concentration to hear me, so you're also breaking your ATP. But when you are calm down, you're doing meditation, for example, or you're calm, your ATP will be synthesized, or you're sleeping, you are not breaking down ATP. So when we are under stress, then we need energy, then our ATP will be broken down. But when we are in non-stress condition, then our ATP will be synthesized. So citrulline can synthesize ATP. It does indicate that it is anabolic metabolism. It is not catabolic. It is not breaking down metabolism. It is producing, synthesizing state of chickens, so which is afforded by the citrulline administration. And what about broiler chicken? It is almost 4 weeks old, body weight 1.5 kilogram, which is commercially very important, but very susceptible to heat stress. So we have found that if we inject them twice before exposing to high temperature, their body temperature declines. This is the control group. Body temperature declines by citrulline. The normal room temperature, high temperature, 7 degree high, the high temperature 30 degrees Celsius. The water injected group body temperature increase like this, but citrulline injected group body temperature increase, but not that high. So this is called thermotolerance. They can survive under high temperature due to the citruline's effect. And some other parameters in the blood, like lactic acid, increased. Lactic acid from the glucose, it can be produced, and I think citrulline stimulated lactic acid production. And lactic acid has a defense mechanism against alkalosis. Under heat stress, chicken do panting, because panting means that they reduce more carbon dioxide from the body, and this causes some alkalosis, which is not good condition for the chickens. So this alkalosis is minimized by lactic acid. So citrulline is enhancing this situation. So citrulline is helping to reduce alkalosis and also producing more ATP. Then some metabolic hormones, T3, thyroid hormone. I think nowadays many people are suffering with thyroid problems. The thyroid hormone is a very important hormone. It regulates different metabolic pathways in our body. And then when we have stress, our metabolism may increase. So as you see here in the black bars, so the high temperature significantly increased T3 hormone, thyroid hormone. But citrulline administration under room temperature and high temperature reduced significantly, as you say, less than 0.01 p-value. So citrulline reduced the metabolic rate. So it does indicate that citrulline can reduce body temperature, which is very important to survive under high temperature. Another thing I like to explain about the insulin. Insulin was increased by citrulline under high temperature. So insulin is an anabolic hormone. It helps to synthesize different things in our body. When you take food, for example, our pancreas secretes insulin. Otherwise, glucose cannot penetrate into the cell to produce energy. So citrulline is helping to increase insulin and thereby help to produce more ATP. And then we wanted to know whether we are taking the citrulline through the oral administration, whether it is entering the brain or not, because brain is the center where body temperature is regulated, as I mentioned previously. So in this case, we just 2 hours time we examined control temperature 23, high temperature 30 degrees Celsius, 4 weeks old chickens, broiler chickens, and we have found that all 3 amino acids, citrulline, arginine, ornithine, with administration of citrulline, arginine, ornithine, all increased in the blood, and simultaneously all increased in the blood, brain. So what the amino acids increase in the blood also increase in the brain. In the brain, we have some barrier. All amino acids or all nutrients cannot pass the blood-brain barrier, we call it, cannot pass to the brain. But the citrulline can pass the brain, blood-brain barrier and go to the brain to control neuronal function. So in future, we like to understand more about the neuronal mechanism depending on the citrulline to regulate body temperature. And also we found that dopamine and norepinephrine metabolism, these are also regulated by oral administration of citrulline, and this has contributed to body temperature regulation, we think. And this was reported in this Frontier of Animal Science journal. And last one is about leucine. Leucine amino acid, another amino acid, essential amino acid, is increased by heat stress, 5-hour heat stress, 40 degrees Celsius in the chicks. But when we incubated eggs, in the hatching time, for the hatching purpose, we incubate fertile egg, and it is 37.5 degrees normal temperature, but we gave little high temperature for 6 hours per day. And we found that leucine decline in the brain of the fetus or embryo, not fetus, embryo, and the brain and liver. So we wanted to know why leucine declined by the high temperature. So injected around 5 milligram of leucine in the fertile egg, and then chicks hatched, and with sexing was done, male and female separated, reared, isolated, and exposed to high temperature for 3 hours, 35 degrees, and control temperature was 30 degrees Celsius, 28 degrees Celsius, I think, sorry. And then female didn't respond to this treatment, but male responded to this treatment. Control temperature here 28, high temperature 35, blue bar water injected, and red dotted bars are leucine injected. You see that with the leucine treatment under high temperature, body temperature is less than the control group. So it does indicate that leucine treatment afforded thermotolerance. They are more stronger under high temperature. And the food intake also declined. This declining of food intake is helpful under heat stress, because when we are under heat stress, if we take more food, it will produce more heat and it will be harmful for us, for animals. So this is a very good effect of in vivo injection of leucine. And commercially important is that during the high temperature, the summer stress, body temperature declines. But compared to the control leucine group, significantly they afforded high body weight gain. So body weight gain reduction due to heat stress was minimized by the leucine injection in the eggs. So this is a commercially very important findings. And then it was published in the European Journal of Animals, which is a very applicable study in future for developing new technology to minimize heat stress in the farming condition of poultry. So to summarize, for the leucine effect, leucine in vivo injection means injecting the egg, causing to change some metabolism, and this cause some imprinting in the early age, and this imprinting come in the young chicks. Their lipid metabolism changes, and that provides or affords thermotolerance. And then, but for the marketing age time, the lipid metabolism was not changing, but amino acid metabolism changed, particularly GABA increased. GABA is a amino acid which can is a inhibitory amino acid, so it provides some calmness. And all these things cause to, and some unknown mechanisms still we have for the growth protection under chronic heat stress, which has commercial value of this treatment. This would be a new technology, biotechnology, we think, for the poultry farming. This is published in these journals. So to conclude the study, L-citrulline and L-leucine have hypothermic roles to beat the summer heat in chickens. However, further research is needed with L-citrulline and leucine in mammals, including humans, to beat the summer heat. To acknowledge, Kakenhi, Ito Foundation, and Kiekai Research Foundations supported this study. I greatly appreciate their cooperation and support, and thanks everyone for listening my talk. Thank you very much.
Thank you very much, Professor Chowdhury. This is a super interesting topic, and apparently people already have questions. So that shows usually it takes a bit of a while, but here I think people already do have questions. And I think one of the things that you showed, and I think also what I get from the first question is, what are some everyday tricks, applications in the here and right now with those like high temperatures that people could potentially, and of course, since a lot of these things are still like subject to intense research, and it also remains to be seen how applicable it is to other mammals, to mammals actually, and also like to humans especially. But from the first question, it seems citrulline seems lemon-containing component. Lemon or orange are good for heat stress. So this would be like a question, and I want to expand it beyond that. So whether it's good or eating lemons or oranges or citrus fruits, but is there anything like either preliminary or so that people could do in their everyday lives through nutrition to potentially deal better with the current high temperatures and potentially also future high temperatures under climate change? So something that they could already right now implement to deal better with the heat-related stress.
Thank you very much for the question. And I think, yes, lemons or orange, they're citrus fruits. They have vitamin C, high amount of vitamin C. So vitamin C and citrulline has a different function. But citrulline, of course, vitamin C, it was found that the vitamin C can increase heat shock protein. Heat shock protein is a protein which protects our cell under high temperature. So vitamin C is important to take to survive under high temperature. But here, I try to explain about citrulline. Citrulline is available in the watermelon and some other like cucumber and some other fruits. Citrulline amino acids available there. And you know, nature provides watermelon during the summertime, not in the winter time, because nature is trying to support us. So we are because part of nature. So citrulline and some maybe melon, this sort of fruits coming in during the summertime to provide citrulline for humans or other animals, so that they can have more citrulline to keep their body temperature under control, not to increase more. So in that sense, I think this sort of fruits would be helpful.
Mm. So it goes, you kind of already answered the second question because it was about eating watermelon. But do you think that, for example, taking vitamin C supplements, or like in this case, as you said, like there's a difference between citrulline and vitamin C. Do you think that taking that in the future, for example, as a daily supplement, as a nutritional supplement, that is even possible? And would it yield the same benefits that like the consumption of watermelons or like citrulline in your research? Do you think that can be a future way of making people more resilient against heat stress, for example, a daily supplement of citrulline. I'm not sure if that already exists, but if we just take a certain prescribed quantity of it, then our body would potentially be a little bit more resilient under high temperatures. Do you think that is something for the future?
So citrulline administration, if we say for chicken first, so citrulline even not permitted to add in the chicken diet at this moment as per the rule of the Japan poultry diet standard. Citrulline is not permitted to add. So we are doing this study and found that citrulline has some many types of beneficial effect under high temperature. So we are trying to promote citrulline. Maybe in future, citrulline will be considered as a summer, amino acid to adding in the summer food. Why citrulline was not considered? Because arginine is an essential amino acid. Arginine can produce citrulline as a byproduct. So arginine is giving in the chicken diet, but so that people are thinking that citrulline is becoming, and so it is not needed. But we have found that if we put citrulline, and some other researchers also report that instead of giving arginine, if you give citrulline, it produces more arginine. So this is for chicken. And another question for human, whether we can have some rules or some regulations or some system in future for the heat stress resilience to have regular some supplement like citrulline or lemon or something like that. So being an animal nutritional physiologist, I cannot take the responsibility to recommend for human at this moment, because we have to do further work with some mammals like some cats or dogs or pigs, cattle, that sort of things, and even some maybe lab mouse or rats. After getting a good result, then we will like to recommend those for humans as well.
So what kind of other like foods or things, like, at least from the time being for right now, and I know like, yeah, it relates to your research, but has there been any other be it from like traditional or so, has there some substances that you want to explore further in the future in your research that has already maybe demonstrated, for example, with people who live in hot climates, such as the Middle East or so, and where they have more natural resiliences, and where maybe local food or so has proven to provide some like heat stress lowering functions? Has there been already something on your radar?
So I think you may know that in Japan, long time ago, people are taking pickles from the watermelon rind, which is a cover. Normally you waste it. We consume the flesh and waste the cover, which is called rind. So rind, we found that some researchers found that a rind containing more citrulline than the flesh. And we conducted some study with the juice of rind juice, and we found that it has strong influence of increasing blood citrulline level, arginine level, and it also reduced blood body temperature in the chicks. So in Japan also, long time ago, people are taking rind as a pickles. And in some country, currently also they take this rind with vegetables. So such items, I think people are taking without knowing its effects of citrulline or something like that.
So I'm wondering if there's any more questions, because I do have a lot, but if are there any more questions at this point in time from the audience? So I just want to make sure. So I do have another question in terms of you said the chicken does not have sweat glands, and I think other animals like dogs also do not have sweat glands. If I am so what would be the natural progression right now on moving on from chickens? Would it be immediately human testing, or what would be an intermediate stage in the process of finding out whether any of this research can also be more or less applicable to humans, or are there already preliminary studies that show, for example, people, as you say, like in the past, they have been consuming citrulline without necessarily knowing that they did. So is there anything like, as an intermediate stage, moving on from the chicken to, like, for example, other types of animals that, how do I say, come closer and closer to human conditions?
So, because people think that chickens are far from humans, but as I mentioned with the Nature paper, chickens' genome, arrangement is almost very close, not same, very close to human than even the rats, mouse, rodents. So, and for my own experience, I did study with quail birds with some brain neuropeptide, which was first discovered in quail. Later, with lab study, it was found that it is existing in amphibians, existing in rodents, existing in monkey, even finally found it exists in human. So some important neuropeptide or important biological molecule that persist through the evolutionary tree from the lower animals to higher animals. So the findings which we are getting from the chickens, we think that not all, some of those findings can be applicable for humans in future. But of course, we like to do some mammals experiment before applying to human.
And speaking about, you mentioned the frog. You showed a picture of a frog in the beginning, and you said, you mentioned again now, like amphibians. So since a large, and my research, so that is why I'm asking personally, since my research deals a lot with biodiversity, do you think some of this research that you're now carrying out could be utilized in terms of saving species, for example, reptiles or amphibians, who are very susceptible to climate variance, and also especially climate change and increasing temperatures? So, for example, would it be possible at some point to develop nutrition or supplements that could be administered to amphibians in order for them to make them more resilient in the long term, for example, through artificial feed, or like supplementing, like, what occurs in their natural environment, but allowing them to face or like at least become a little bit more resilient? Do you think your research could be applied through, for example, ingestion of, let's say, heat resilient increasing substances?
Yes, this is a very, very good question, because amphibians are very close to the living, the habitation is very close to the earth, and the surface temperature really affect them very quickly. So I also did my study 1 year with using bullfrog, and then I found that they like korogi, a kind of insect for their food. I always collected that sort of insect for feeding my experimental bullfrog. And recently I have joined one poultry science conference in Kyoto, and then I found that feeding this insect, korogi insect, in Japanese they call korogi, this insect can decline a certain enzymes that is called alkaline phosphatase. Anyway, the certain enzymes, which enzymes is responsible for cell death. So the insect containing some substance which help to increase the cell survival. So increase the betterment of the frog. So, for example, our citrulline treatment can increase the citrulline content in the meat of the chicken. So simultaneously, if we can think whether some citrulline-rich meat or some insect can be produced, and those can be fed to the frogs, and then frogs may have more citrulline in their body to survive. But before that, we need to study a little more about the metabolic process of bullfrog, because the metabolic cycle, what I have shown you, that is for chicken. We have to know the similar cycle is also existing in the bullfrog or not. If they are different, then we have to think in different amino acids or different way.
Yeah, thank you very much. It shows just the complexity of things, but your research is very relevant and very interesting from a point of view of just generally trying to make species more resilient towards heat and heat related stress, and also heat related lethality. So that is something that I think your research will hopefully provide more answers in the future of what could be some pathways. And then I know someone just thanked you for your research. And with that, I would also do the same, because we are unfortunately already at the end of today's presentation. The plethora of questions also from the audience shows this is something that affects everyone, and I think everyone would like to see solutions of how they can make their bodies a bit more resilient. So thank you very much for your time today, first of all, from the entire QAOS team. And we would also like to thank you for explaining a little bit, contextualize a bit. And with that, I would also invite everyone who's interested in this topic to visit Professor Chowdhury's website to read his papers, and ideally reach out to him if you have further questions, or if you're very interested in this topic, or so please do reach out. And then with that, I will now hand it over to the QAOS team to talk a little bit more about future brown bag seminars. But I would also like to one more time thank Professor Chowdhury for today's presentation. Thank you very much.
Thank you, Professor Smaha. Thank you very much for the nice emceeing. Appreciate it.
ルイ先生、シュマハ先生、ありがとうございました。最後に事務局よりご案内させていただきます。まず、次回のブラウンバックセミナーのご案内です。次回は来週10月2日水曜日12時10分より、九州大学未来社会デザイン統括本部の岡田映像教授より、椅子の歴史に学ぶ社会のデザインと題し講演を実施いたします。詳細はホームページをご覧ください。